Yeah, so my name is Tae-hee, third-year medical student at the University of Wisconsin School of Medicine and Public Health. So we're going to talk about aging brains and how neurodegenerations can affect this the normal aging. So now we're talking about abnormal aging. So before we start, let me just briefly introduce myself. So before joining med school, I did the PhD training and postdoctoral research at the Department of Neurological Surgeries. So I did the PhD in neuroscience, and this is Dr. Dempsey, who is the chair of the neurological surgery department. And this is my mentor, Dr. Raghuvemuganti, and then this is my research team. All right, so now we're going to talk about normal aging within the brain, and how this abnormal brain can be compared to a normal brain. So if you remember last week's talk, Ahmad talked about brain kind of shrinking in terms of the volume. So we call it as brain atrophy. So this can be happen. So there's a number of reasons that can cause this shrinkage of this brain. So this is more like a healthy, normal kind of a brain of the young individuals. And this is kind of old, elderly, the post-mortem elderly brain. And it is okay, and it's kind of normal to see a shrinkage of this kind of volume, and then you see more of this kind of the gap between the brains. But this is normal, but what is abnormal is that there's more gaps between these kind of the brain, the ridges. And then another thing, if we look at the kind of molecular levels, you're gonna see accumulation of these proteins. So proteins are really important. First of all, it is a building block of our muscles. So that's why we eat proteins, so that we can build our muscles. But also lots of components of our bodies to maintain our normal brain functions, like kinases, there's the channels, there's a bunch of stuff that is made up of proteins. And then, if something goes wrong, maybe it's because of the oxidative stress, like you breathe a lot, and then that can increase this oxidative, reactive oxygen species, or something happens. We don't know exactly what it is. The research is still going on. That can cause this abnormal accumulation of the proteins. It could be amyloid, in context of Alzheimer's disease. In Parkinson's disease, we're going to talk about this thing called Lewy Bodies. But you're going to see the common theme of these all neurodegenerative conditions is the accumulation of these abnormal proteins. All right, and then when that happens, you're going to lose the connections between these neural cells, the brain cells. And that's probably because of the loss of these neurons within the brains, because of some trauma, or because of some sort of insult, or because of this accumulation of these abnormal proteins that interferes with communications between these neurons. So some degree of this loss of connection is normal again. That's why we see some memory problems. We see some movement problems. That's kind of normal because everyone does that. However, if it is like out of control, then that is abnormal. And some research is showing that this accumulation of abnormal proteins, it's again somewhat normal. We do see some degree of amyloid plaques in our brains, even if we don't have Alzheimer's disease. We do see some degree of Lewy bodies in the brain of normal, healthy brains, even if we don't have Parkinson's disease. However, if it is too much and if it is widespread, then it can possibly cause some abnormal conditions like Parkinson's disease or Alzheimer's disease. All right, so we just looked at the molecular levels, and if we see just abnormal neurodegenerations or abnormal accumulation of proteins, then we see some clinical symptoms. So as an example, I brought this kind of graph. So this one illustrates the age-related cognitive changes. So the x-axis basically represents ages. So here we're talking about like 40 years old individuals. And then as we move along the right side, you're gonna see 65 years and 90 years old. So now, and then the y-axis right here, higher, the better in terms of cognitive functions. So if you look at the 40-year-old individuals right here, he just finished the marathon, his cognitive function is probably way up here because he's healthy. And then this yellow line right here represents a normal aging. As you can see here, there's some declining in terms of cognitive abilities. And this is not linear. One thing you can tell, as you can see here, it's kind of gradual declining, and there's some incidence, something happens. It could be infections, it could be a trauma. That kind of significantly accelerates the decrease in terms of a normal aging, and then it gets back to the normal rate of declining. But one thing that I want to emphasize is that this process cannot be slowed down. Maybe a little bit, but this is inevitable. So again, this is normal. And then some people, they maintain their 40-year-old cognitive ability. So we sometimes call it super aging. Probably it's because of the genetics, I don't know, or maybe the environmental factors, what they do, maybe multifactorial, but we don't know about that. So this is a super aging, and then let's say, man, I think I have some memory issues right now, so maybe I should go and talk to my normal doctors. You go in, and then the doctor's gonna do some testings, and they said, yeah, there's some issues with memories, but they always ask these questions. Okay, so how about, are you okay with doing your activities of daily living? Are you still able to take care of yourself? Taking showers or doing laundries? Can you still drive around your town? Can you navigate through your neighbors? If you can do that, then probably, yeah, I can see that there's some memory issues, but it's still kind of okay. So the threshold that divides between kind of mild cognitive impairment and abnormal aging of kind of dementia kind of thing is whether you can still maintain your activities of daily living. If you cannot do that, that's pathologic. That's something that requires some medical intervention. If you can still do that, then probably we can keep an eye on that and then see whether there's accelerations in terms of memory declining. And we still don't know what makes someone having like mild cognitive impairment versus dementia. Probably there's some molecular components behind it. Something like we just talked about. Maybe there's a more accumulation of these abnormal proteins. Or maybe the brain is like losing more cells. Right, so and then this just illustrates lots of things that are happening inside of the brain. We talked about brain atrophy or brain shrinkage. And then one thing I want to point out is like this cerebral microbleeds and then silent brain infarctions. So microbleeds. Some bleeding's happening inside of the brain. Maybe it's okay. And our body, and then nobody really care about like having some bruises on your arm. That's basically happening, a bleeding inside of your bodies. But nobody really takes that seriously because our body has our natural kind of a clearing system so that they can take care of themselves. So a little bit of bleeding is okay, but if it is too much, or if it is pathologic, like brain hemorrhage, that's the problem. And then this is something that I kind of learned while doing this research, but this Parkinson's disease and any other neurodegenerative conditions, this abnormal accumulation of iron and calcium dysregulation is kind of associated with all this Parkinson's disease and Alzheimer's disease. So maybe they're kind of responsible for the pathogenesis of all these neurodegenerations. And then silent brain infarctions. So infarction is kind of fancy word for brain death, brain cell death by loss of blood supplies. So silent brain infarctions, probably you know this better in terms of mini strokes. So our research is showing that probably if we get older, we have this mini strokes every once in a while. And then I think this mini stroke as in this analogy. So what doesn't kill you makes you better, makes you stronger. So there's the line of this research in basic science. It's called ischemic preconditioning. So what they do is that, so they grow culture this neuron cells in the petri dish, and then you typically give this cell culture medium, which has nutrition and oxygen. But then you take away to replace that culture medium with no nutrition, and then put it in the chamber where it has no oxygen in it. So basically it mimics the ischemic stroke environment. So if you leave that for, say, typically like 1 or 2 hours, 80% of cells die. Makes sense, right? Because there's no oxygen, no nutrition. But if you just expose that environment for like 10 minutes and then take out the brain, maybe 95% of cells are still alive. But if you subject the same cells who had that brief kind of ischemic exposures, and then you're gonna expose those cells to a subsequent actual 2-hour-long ischemia, those cells actually survive much better. So this is the idea of this ischemic precondition. You're basically precondition these cells to an ischemic environment very briefly, kind of sublethal level. But then when you have actual ischemic insult, you're actually more resilient. Maybe those cells, when they're first exposed to this ischemic conditions, they express like a survival, kind of pro-survival molecules, so that when they actually have this ischemic insult, they're doing much better. So my idea is that maybe depending on the severity of this silent infarction or mini stroke, maybe it's beneficial when you actually have a stroke. But of course, if it is too severe, then it can actually cause brain damage. So the research is showing that these mini strokes, probably this is kind of normal processes, but our body has our own kind of a defense mechanism so that you don't actually see much of a damage or clinical symptoms. Right, let's move on. And then there are lots of brain regions that is implicated in the abnormal aging or neurodegenerations. So this diagram is brought from the Alzheimer's disease field. And then, although this is like specific to Alzheimer's disease, these brain regions are typically implicated in any kind of a neurodegenerative condition. So we're talking about hippocampus, highly important regions for learning and memories. If you damage hippocampus, you just cannot consolidate memories. So what happens if I say that, please remember the number 41265, and then you're just remembering it. So that requires working memory. That's somewhere in front of your cortex right here to maintain that information. But if you wanna convert that short-term memory into kind of long-term memory so that even 5 days later, when I ask you, you can just recital by heart, that requires hippocampus. If hippocampus is damaged, you just cannot do that. You cannot convert the short-term memory into long-term memory. So it's highly important. And then in Alzheimer's disease, we see those abnormal plaques all over the place within the hippocampal area. Amygdala, very important for emotional regulation, especially in the context of fear regulation. So there's this animal studies. So rats, they naturally scared of all these snakes. If you damage the amygdala in rats, and then they let them just kind of foraging outside, and then if they encounter the snakes, they don't fear. They just still do whatever, they just mind their own businesses. So it's pretty interesting that just how amygdala regulates the fear conditioning. And then there's the cerebral cortex. Amon last week talked about the cerebral cortex is what makes human human. So if the cerebral cortex is damaged because of this abnormal plaques or loss of these brain cells, yep, you're gonna see personality changes, you're gonna see the impairment in the executive functioning. That's exactly what happens to Alzheimer's disease. All right, so we just kind of briefly overview what's really happening inside of the brain. So now I'm gonna use a specific examples to talk about why am I emphasizing this abnormal accumulation of plaques or proteins. So we're gonna use two examples, Parkinson's disease followed by ischemic strokes. The reason I picked these two conditions is because both of these conditions affects your movement. And they are, if you put on these two conditions in the spectrum, they're on the two extreme ends in terms of severity and then disease progression. So Parkinson's disease, it takes years or even decades to kill the brain cells. Ischemic strokes, time is brain. It takes hours, even minutes, to kill these brains. In fact, once the blood supply is cut off, every minute you're losing 4 million cells. So I'm literally saying time is brain in case of ischemic stroke. All right, let's talk about Parkinson's disease first. All right, so what is Parkinson's disease? It is a progressive nervous system disorder, mainly affecting your movement. We're gonna see 4 cardinal symptoms of Parkinson's disease right after this. But this is the disorder of movement, at least at the beginning. Once the disease progresses, you're gonna see some other changes, like personality changes, and you're gonna see psychotic features like hallucination, things like that. But at least at the beginning, it is the disorder of movement. And there is a gradual, as I said, cells don't die within minutes or hours. It takes years to kill these brain cells. And then initial symptoms may be barely noticeable. Maybe you have just a little bit of tremor in your hand, and I don't know, maybe this is kind of really annoying, but it's not like, a big of a deal. But tremor is a very common presenting symptoms of Parkinsonian patients. And also there's other symptoms like stiffness or slowing of the movement. And right now, once you have Parkinson's disease, there's no available cure. There are medications and implants that can manage symptoms, but there's no cure for this. All right, so what are those 4 cardinal symptoms of Parkinson's disease? So the most common presenting symptoms of Parkinson's disease is this tremor. So tremor is a resting shaking of rhythmic shaking of arms, legs, or chin, but typically you see this tremor in your hand. And then this is typically one-sided. And unlike essential tremor or functional tremor, where you see kind of shaky when you feel nervous or kind of stressed, those are typically in both hands. It's bilateral. This one is typically unilateral, one-sided, and unlike this functional tremor, this is kind of high amplitude. You see like this kind of obvious shaking. So as you can see here, this patient has this typical tremors. And then another kind of a characteristics of this Parkinsonian tremor is that it happens at rest. So we also often call this Parkinsonian tremor resting tremor. The second is bradykinesia. Brady means slow, kinesia means movement. So it literally means slow movement. So as you can see here, I have some examples here. First, I want you to pay attention to the posture of this patient. It's kind of stooped, right? It's not like, upright, it's kind of stooped. And then it's making very small steps. And second, there are some tremors right there. And especially in this patient, you can see it appears that this patient kind of forgets kind of unconscious movements like swinging arms when walking. So this is typical characteristics of Parkinsonian patients. They just forget this unconscious movement. So bradykinesia, they are slow. Third, rigid muscles. So when you test this Parkinsonian patient's muscle, it's very rigid. You're gonna ask the patient, can you kind of completely relax your arms so I can check your muscle tones? If non-Parkinsonian patients, they can relax, and then if I just wiggle the arms, of course, it's very fluid, right? But if the patient has Parkinsonian symptoms, it's so rigid, as if you're actually turning the cogwheel. So sometimes we describe this Parkinsonian patient symptoms cogwheel rigidity. The last symptom is instability. So you lose postural reflexes. So for this, in the clinic setting, doctor's gonna ask you to walk in tandem, like heel to toe. And actually it's pretty difficult if I just try to do that. I feel this is kind of hard, and then I can't even imagine if you have this posture instability, it might be even more difficult for them. So these 4 cardinal symptoms, bradykinesia, resting tremor, posture instability, and the cogwheel rigidity, you need all 4 of these at the time of diagnosis. Right, and there are some other features of Parkinsonian symptoms. You have some difficulty initiating movement. You lose like unconscious movement, such as swinging both arms. And then they have some speech difficulties. They tend to speak more softly, quickly, and their speech is kind of more slurred or hesitate before talking. And their tones are pretty flat. And also there are some writing changes. They tend to write smaller and kind of scribble, as you can see here. So this is like typical Parkinsonian micrographia. And it appears that this patient has some sort of therapy, writing therapy, and that is much improved compared to this. Right, so now, yes?
Is there any physical therapy that could help with any of those movements?
Yeah, absolutely, yes. There's a physical therapy specifically designed for improving Parkinsonian movement symptoms. So yeah, so your doctor's I was gonna get to that point, but once your diagnosis and then talk to your regular doctors, they're gonna refer you to movement disorder specialist. They're gonna officially diagnose you for Parkinson's disease, and then they're gonna refer you to like PT, OT, and like speech therapy, and all these things. And then your regular doctor's gonna manage your medications, along with like neurologist. And then you also have to work on lifestyle modifications also. So that's basically the idea of the Parkinson's disease management. Right, so the real question is, why are we seeing these symptoms? There must be something going on inside of the brain. That's why we're seeing this, right? So there are a lot of things we still don't know in terms of Parkinson's disease, but this is something that we know. So Parkinson's disease, all these movement symptoms, is caused by loss of dopamine producing neural cells in a very specific brain regions called substantia nigra. So substantia nigra substantia meaning substance, nigra meaning dark. So basically this literally means dark substance. The reason is, first of all, this substantia nigra is located in the deep inside of the brain, right, this red area, so it's pretty deep, right in the middle. And as you can see here, this is a normal, healthy substantia nigra. Can you see this dark band right here? These dark bands is basically all this dopamine-producing neurons. When you look at the post-mortem Parkinson's disease patient's brain, you don't see this dark brain as much as the healthy brains. So this is number one pathological hallmark of Parkinson's disease. Clinical symptoms, those four clinical the symptoms are basically clinical hallmarks. This is the molecular pathological hallmarks. So this means that if we just take this brain to a pathologist, not telling them this patient when alive had Parkinson's disease, they're going to examine the brains and then, I think this patient had a Parkinson's disease because, first of all, they don't have these dopaminergic neurons in substantia nigra. I'm going to talk about the second one, but this is the number one pathological hallmark of Parkinson's disease. And this dopaminergic neurons from substantia nigra kind of projects its extensions to other part of this brain regions called striatum. So that's why the researchers named this kind of projection pathway a nigrostriatal pathway. So basically, to summarize, Parkinson's disease is caused by impairment in this nigrostriatal pathway. And that's because of the loss of dopaminergic, the brain cells in substantia nigra. All right, this is another picture. It's kind of more clear. This is healthy patient. This is Parkinsonian patient. Again, substantia nigra right here. Yep, dark bands right here. Looks pretty healthy in terms of substantia nigra. You lost all those dark dopaminergic neurons. And you can kind of thin slice this brain tissues and stain them for specific markers. So in this case, what they did is they just kind of very thin slice this brain, and then they stained them with all these brain cells. So you can see here, these are all dopamine-producing neurons. Looks pretty healthy to me. And then, as you can see here, you don't see these cells here, maybe one or two, and then to me, it doesn't look like these are as healthy as these cells. First of all, the cytoplasm, the inside of the cell is pretty faint compared to this. It's dark here, so it's more dense. Here, I don't see they're very healthy. And another thing that is very interesting that the researchers found is that if you examine this Parkinsonian substantia nigra, you see this little things that you don't typically see in the healthy brains. And then researchers think, oh, what is this? And then the first person who discovered this is Dr. Lewy. So they named this as the Lewy Bodies. So we just collected several clues. Parkinson's disease is caused by loss of dopaminergic neurons in the substantia nigra. And then at the same time, when you examine this substantia nigra, you see this little things, kind of abnormal protein aggregate called Lewy bodies. So this is number two of pathological hallmarks. Number one is dopaminergic neuronal loss in substantia nigra. Number two is Lewy bodies. Right, and then when later researchers analyzed, okay, what is this Lewy bodies? And they found that there's some other things, but a major component of this Lewy body is called alpha-synuclein. This protein called alpha-synuclein. We're gonna get back to this later, but I want you to remember this protein called alpha-synuclein. That's a major component of this Lewy body. Right, and I just put this slide to show that, yes, substantia nigra is right here, but this Parkinson's disease, it doesn't start from where the substantia nigra or striatum is. It starts from the way below at the brainstem levels. And then it takes years to get to this point. And then once it reaches to where the striatum is, then you're going to start to see these clinical symptoms. So even if you start to show this kind of fine, subtle tremors and some slowing of the movement, probably the degeneration has happened for many, many years. Okay, so we just talked about some of the pathological hallmarks, loss of dopaminergic neurons in substantia nigra, presence of Lewy bodies. So Lewy bodies is basically a clump of specific substances within the brain cells. So, and the major component of this Lewy body is alpha-synuclein again, a protein called alpha-synuclein. Abnormally misfolded and pathologically clumped alpha-synuclein proteins. And then what is alpha-synuclein? Why is it in the Lewy bodies and they're causing this Parkinson's disease? First of all, we don't know whether alpha-synuclein causes Parkinson's. $1 million are spent to understand that part right now as we speak, but still the mystery is out there and we don't know exactly what's going on. But what we know is that about 1% of total proteins inside the brain is made up of alpha-synuclein. So it's not just localized in the substantia nigra, it's just all over the place. And when researchers are trying to understand, okay, what is the normal function of this alpha-synuclein? Why do we have this? If it is just widespread, it must be important, right? So when they studied it, it is involved in this release of dopamine neurotransmitter. And dopamine is really important. If you erase dopamine completely from our system, you're gonna die because this is critically involved in the normal functioning of our daily lives. So we thought, okay, maybe alpha-synuclein is really important. But if you completely remove alpha-synuclein from the system in genetically engineered mice, these mice can survive, meaning that this alpha-synuclein can be dispensable in the life. So there's a mystery out there, still trying to solve what exactly is alpha-synuclein is doing. But so far, it looks like alpha-synuclein is kind of involved in the release of this dopamine neurons. And found in all Lewy bodies, a major component, about like 90% of this Lewy body is made up of alpha-synuclein. So why is $1 million questions? Why is alpha-synuclein inside of the Lewy bodies? And what is it doing? Is it beneficial? There's two lines of thought. So Lewy bodies is present in the Parkinson's disease. Is it because Lewy body is causing harm to kill this dopamine producing neurons? Or is kind of a protective mechanism. So let's say if alpha-synuclein is really toxic, this Lewy body is probably kind of sequestering this alpha-synuclein so that it cannot spread kind of further. So it's more like a protective mechanism, like kind of putting barriers around the alpha-synuclein. So there are lots of different theories going on, and based on those theories, researches are going on, but still jury's out there. Right, so there are some other causes. If you look at the Parkinson's disease, majority of cases are because of just happens, like we don't know why it is happening. So we call it sporadic Parkinson's disease. We don't know what's causing it. They don't have any risk factors besides typical ones like age and sex. But about 10 to 15% of this alpha-synuclein is caused by genetic mutations. So there's several genes that are identified that can cause early onset familial or genetic Parkinson's disease. And as you can see here, one of those genes is alpha-synuclein, meaning so alpha-synuclein is not that long protein, it's about 140 amino acid. If you just mutate a single amino acid, that can cause basically a Parkinson's disease. It's pretty scary actually. So just one amino acid is disturbed, it can cause Parkinson's disease. And there are some environmental triggers that are associated with Parkinson's disease. So for example, 6-hydroxydopamine, MPTP, and paraquat is a chemical that is widely used in the herbicide. So these are all kind of associated with Parkinson's disease. And MPTP is kind of funny. So in 1970s, this one grad student from chemistry department, he kind of synthesized synthetic heroin. And then part of this synthetic heroin, he used MPTP as part of it. And then like 3 days later, so he self-injected that heroin. And then, 3 days later, he showed all sorts of this typical Parkinsonian symptoms. And then later, the researchers figured out that there actually this MPTP that is specifically targeting those dopamine-producing neurons in the substantia nigra. So naturally, researchers are using this MPTP for Parkinson's disease research right now. So you just inject it, and somehow it specifically, like selectively targets those dopamine-producing neurons. So this is a really valuable research tool, along with paraquat also. So we just talked about dopamine a lot in the context of Parkinson's disease. So dopamine is really, really important. So this is, first of all, when dopamine is released, the recipient neurons that receive dopamine, the dopamine excites that neuron. So this is excitatory neurotransmitter. And it is produced in several places. Substantia nigra is one place. There's another place called ventral tegmental area, VTA. The name is not really important. But as you can see here, those things project all over the brain, controls the cortex, again, cortex executive functioning, and other things that makes human, and also the movement when it goes to the striatum right in the middle. So now this is the nigrostriatal pathway we just talked about. And this is the cortex. Again, dopamine is very important. And if you look at this, dopamine is highly involved in learning and memory, motivation. In fact, let's say I really have to finish all these chores, but you don't have motivations, right? That's probably because your dopamine level is kind of low. Dopamine makes you feel, I want to do this. I want to do this. That feeling of motivation, that's caused by dopamine. But let's say once you accomplish whatever you planned it, you have this sense of accomplishment. That's not dopamine at all. That's more like heroin. That the feeling of hedonia is caused by heroin. But I kind of digressed a little bit. And other physiology is also controlled by dopamine, like lactation. Your mood is controlled by dopamine. Attention. In fact, ADHD, there are two medications. You probably all know Adderall. And then there's another one, methylphenidate. That is actually treat the ADHD symptoms by increasing the dopamine in our brains. So dopamine can control attentions and movement. We just extensively talk about dopamine in terms of movement. So this is a pretty neat video, actually. Let me see if I can show this. So basically what they did is that, this is kind of very early studies. They placed this electrode in one of the brain area called lateral hypothalamus. So lateral hypothalamus does not directly release dopamine, but it indirectly stimulates this VTA that I talked about, one of the dopamine producing centers. So, and then what they did is that they just placed this lever right here. So rat can actually self-press this lever so that they can kind of stimulate that LH. So let's see what happens if rats can control the stimulation of that LH.
The video clip shows a rat with an electrode implanted in the lateral hypothalamus. The animal can freely move about the cage. By accident, the animal presses the lever. As a result, a weak electrical stimulation is passed through the electrode. The electrical current activates the reward system, which in turn reinforces the rat's behavior. The rat presses the lever continuously at a constant rate, at the expense of other vital behaviors such as eating and drinking.
So again, motivation. Right. So next question is, can Parkinson's disease be prevented? All right, so if you look at the risk factor of Parkinson's disease, age. Older, the more chances of getting Parkinson's disease. Sex, males tends to get more Parkinson's disease than females. Family history, yep, there's a family history component right there, about 10 to 15%. History of toxin exposure, yep. Like Paraquat, MPTP that I talked about, that is associated with Parkinson's disease. And then this is more like a recent one, history of REM sleep disorders. So the sleep has different stages. There's like light sleep, deep sleep, and then there goes REM sleep. So REM sleep, during this REM sleep, you typically have dreams. And then because some of the dreams are pretty wild, and then there's a potential risk of you acting out. So our brain kind of releases this muscle paralyzer so that you're completely paralyzed whilst you're on a REM sleep. But some people, they don't get, their muscles are not paralyzed, and then they actually act out while in the REM sleep and having dreams. And so we sometimes call it dream enactment. If someone are at the right age and then sex, and then have a history of this dream enactment, the new research is showing that there's a more significantly increased risk of having a Parkinson's disease. So why am I talking about this when I talk about prevention? Age, can you change it? Sex, family history. There's so far all these identified risk factors are non-modifiable risk factors. You cannot do much about it. So we don't know what causes Parkinson's disease. We know that these Parkinson's movement disorders are caused by loss of dopaminergic neurons there in substance nigra, but we don't know what actually causes that loss of dopaminergic neurons there. So because we don't know what's causing it, we don't know what actually prevents, losing those neurons. And there's some preclinical research going on saying that, regular exercise can prevent it, and then drinking more coffee is associated with Parkinsonian symptoms prevention. And then, as I mentioned, the iron accumulation in the brain is somewhat associated with Parkinson's disease. But again, there's no clinically proven method to prevent this Parkinson's disease. Okay, so we don't know, and we don't know. Yeah.
You mean too much iron?
Too much iron in the brains, yes. All right, so what about the outlook of this patient with Parkinson's disease? Yep, as I said, we don't currently have a way to prevent this Parkinson's disease. But once you have a Parkinson's disease, you're going to talk to your doctor. As I said earlier, you're going to see a movement disorder specialist. They're going to manage medications and going to refer you to PTOT and then speech therapy. And also, there are other kind of implantable devices that can kind of control this Parkinson's disease symptoms, Parkinsonian symptoms. And then you also have to work on improving lifestyle modifications like doing exercise, healthy eating, improved sleep. And with the right course of actions, many patients with Parkinson's disease can live fulfilling lives. So it is still possible that, chances are with the right things you're gonna do, you're gonna die with Parkinson's disease. Parkinson's disease is not gonna kill you. So that's the idea of it. And then I'm just gonna briefly kind of mentioning about this deep brain stimulation. I'm not gonna talk about how this is done or what's the therapeutic efficacy. The reason I'm showing is that this is a pure result of years of research on Parkinson's disease. The reason is that because researchers understood what's the pathway of this dopamine-producing neurons. We just talked about nigrostriatal pathways that controls movement. So we know that because dopaminergic neurons are all lost, we know which brain regions are kind of understimulated in Parkinsonian patients. So you're gonna carefully place this electrode inside of the brain where it is understimulated. And we can control this activity of those brain regions by placing this electrode, and then this is basically a pulse generator right here in your chest. And then I feel like this is pretty dramatic. So this is a Parkinsonian patient without DBS, this device on. And then, as you can see here, she's showing all sorts of like Parkinsonian symptoms, like tremors, and then have a posture instability, still postures, everything. And then once this machine is turned on, he's walking like that. And then I thought for many, many years, I thought this is kind of a dramatization of this device. But I actually saw, I had a couple of days spent in the movement disorder clinics, and then I saw those patients like coming into a clinic for like change the batteries of this DBS machine, and then so they have to turn off the machine for a while, and then immediately you can see this patient having like resting tremors and then all sorts of like Parkinsonian symptoms. When this machine is back on, it's completely normal. So I thought, wow, so this is actually working. Yeah, so what about the future of Parkinson's disease? So since Parkinson's disease is caused by loss of these dopaminergic neurons, the idea is that, okay, what about replacing those lost dopaminergic neurons, right? That's kind of a natural kind of conclusion of those once you understand the mechanism of Parkinson's disease. So there's some research going on using the stem cells. Basically, these are immature cells. Their fate hasn't been determined yet. So you're gonna take out some of the stem cells from our bone marrow or embryos, which is kind of ethically debatable. And then you're gonna determine the fate. So that now you're gonna be dopamine producing these cells. And then you're gonna kind of place it, kind of implant it inside of the substantia nigra. And then yeah, hopefully those cells can produce dopamines, make some new connections and produce dopamines. So that's the whole idea. There's not much of success at this point, but I think the whole premise is kind of promising. So we'll see what happens in the next 10 years. And another line of research is that, okay, these cells are dying. Can we protect these cells? So there are lots of like neuroprotective agents out there so that we can protect these cells. If let's say somehow if we can identify this patient who is kind of vulnerable for future attacks of this Parkinson's disease, can we protect these neurons? So there's some research is going on, not some actually, there are lots of research is going on in terms of neuroprotection of these cells. And one of them is I just found just random things. There are many, many other kind of agents, like just glial-derived neurotrophic factors. It's, at least in the preclinical stage, is shown to protect its dopamine-releasing neurons. So my final remarks on Parkinson's disease is it's not fatal, and early detection is the key to help reduce complications that shorten life expectancy. So if you see something, talk to your doctor and then see if you can improve or manage the symptoms as early as possible. All right, very quickly, so that's a Parkinson's disease. We're talking about years of a process of cell death. Now we're in the far other end of the spectrum. So ischemic stroke, we're losing these cells really rapidly. It takes minutes and hours. And it's caused by rapid loss of brain function because of the cutoff of the blood supply to a brain. And then when that happens, if it is like major arteries, the downstream effect is even worse because the major arteries supplies, let's say, the whole half of the hemisphere of the brain. If it is just a small arteries, maybe just a little bit of damage. So it depends on depending on the arteries that is affected, the severity of the disease can be a whole lot different. If you look at the entire, so stroke, there are two types of strokes, ischemic stroke and hemorrhagic stroke. 87% of all cases of stroke is ischemic strokes, meaning that you lose those brain functions because of the cutoff of the blood. We're going to talk about hemorrhagic stroke in a bit, but 87% is ischemic stroke. And every year, about 800,000 Americans affected by this. And the cost, medical cost is almost $34 billion. That doesn't include emotional cost by the patient themselves and the loved ones. Along with Parkinson's disease, this is the leading cause of disability in the United States. And depending on where it affects your movements, memory, and language. Unlike Parkinson's disease, this is caused by vascular issues. You're blocking your blood vessels. And so there are certain things you can do to improve or prevent that. So age and sex, again, non-modifiable risk factors, you can do much about it. There's a more chance of getting these strokes as you get aged. And if you're male, I think the median onset of strokes in males is like 66 years old, females 70 years. So males tend to get these strokes a little earlier. But to prevent blockage of this blood vessels by blood clots or cholesterol kind of plaques, you control your cholesterol levels. Smoking can increase your blood pressure, hypertension, just high blood pressure, and diabetes. So it sounds pretty similar, right? Like, if you go to see your regular doctors, they always ask, yeah, your cholesterol level is kind of high. And are you taking your blood pressure medications? Have you thought about taking statin for your cholesterol? Yeah, so this is the reason why your doctors basically sing about these guys. Because if you have any of these, chances of having stroke is basically, it's just like triple, sometimes quadruples. So diabetes is really, really scary one if you have it. All right, and then when it comes to stroke symptoms, remember, be fast. So this is the mnemonic for remembering the stroke symptoms. B for balance, one-sided. If somebody's basically stumbles one side, or blurry visions, typically one-sided again, and facial droop, again one-sided. And then A for arms and leg weakness, again one-sided. And speech problems, kind of slurring speeches, or even though the person is speaking, if it is just completely gibberish, again, you have to suspect, you can just laugh at it and then just move on. You make sure to call 911 and go to see an emergency doctor. Right, a T, time to act, call 911. So be fast, remember that. Ischemic stroke, blocking of this blood vessels, and then the downstream brain area is affected, can receive oxygen and blood sugar, so you're basically losing all these functions. Hemorrhagic strokes, blood vessel ruptures. You're losing blood, so the downstream brain areas cannot really receive the blood. And so this is why the hemorrhagic stroke is more lethal. So ischemic stroke, people don't typically die because of ischemic stroke, but hemorrhagic stroke can be very fatal. And just a brief moment about mini strokes. So it is type of ischemic strokes, but unlike ischemic strokes, it resolves by itself. So maybe it lasts a few seconds or a few minutes, and then it just goes away. And as I mentioned, that maybe theoretically, it can be protective for your future strokes, but clinically, this is a medical emergency because you don't, at the time of the symptoms, you don't know whether this is mini strokes or the actual full-blown ischemic stroke. So you have to go and go to the emergency room for this if you have any kind of stroke symptoms. All right, so what happens to the brain when blood flow gets interrupted? So just briefly, what's happening inside of the brain sequentially. Let's say a part of the brain stops receiving blood from basically the blood vessels because cholesterol plaques or some blood clots formed, and then you're not receiving blood anymore. Blood carries oxygen and glucose. When that happens first, our body has lots of components, our cells has lots of components in it, and that requires energy. Now the energy is depleted. I think, if I remember correctly, each neuron, the neuronal brain cells has about 5 minutes of reserve of these energies, or probably maybe less. And once those energies are gone, now you're in trouble. There are certain chemicals that are highly toxic, very important for our normal functioning, but once it is dysregulated and it's just released like uncontrollably, it can be really toxic. One of them is this neurotransmitter called glutamate. When glutamate is just released out of control, it can be toxic. It can uncontrollably excite neighboring cells. And then when that happens, cells eventually die. So we call it excitotoxicity caused by this glutamate. And part of the reason why people think that glutamate is toxic, and then have you heard about this MSG, right? The monosodium glutamate? The part of the reason why people think that this glutamate is so bad for our health is because it has glutamate in it. And then we know that glutamate can cause toxicity, so natural conclusion is that, yep, when the glutamate is, we ingest MSG, it can actually cause brain cell death. So that's a natural thing. But researchers have shown that even if we eat glutamate, it does not like convert like 1-to-1 ratio. Let's say if we eat 100 glutamate, MSG, it does not increase our glutamate inside of the brains. So there is no toxicity between eating MSG and causing this glutamate-mediated toxicities. But that's the idea behind like why people avoid eating MSG. And then mitochondria, a center for energy producing. Now we are not receiving glucose. Mitochondria is not functioning well. So mitochondria is also a place for this release of molecules called cellular signals for cell death. So when mitochondria releases this molecule called caspase, it basically is a signal for cells to basically suicide. And this suicide is quite important for our normal functioning. If you think about, you've probably heard about the analogy that we're actually from our far ancestor was from the fish. And we actually do see the evidence that our ancestor is fish. When we are born and then we're still in the embryo stage, we do have these webs inside of the womb actually. And then somehow our body knows that, yep, this has to go away so that you can be human. So our body sends a signal that, yep, you guys should die so that we can actually have fingers. So the webs are kind of disappear as time goes by. And that's by process of this apoptosis or programmed cell death. So that's controlled by mitochondria. But if something happens and the mitochondria gets dysfunctioned, you can see all sorts of cellular suicide, murder, and oxidative stress. Okay, so when that happens, when the blood supply is cut off, you see cells dying. Obviously, all those, excitotoxicity, mitochondrial dysfunction, other method. And then once cells die, the ground zero is called ischemic core, right there. When cells die, nobody can rescue it. So this is not the area of research that somehow we can kind of revive these cells. Once cells die, it's done. That's why time is brain. You have to go right away. Don't delay it. But the idea is that once these cells die, these cells has all these nasty chemicals inside there. And then as they die, they just dump this to neighboring cells. So let's say because of the blockage of the blood, there's only this much of the cells died so far. But as time goes by, because of those nasty chemicals from the dying cells, these cells are in danger. Once they receive those like glutamate and other like caspase that program cell death molecules, these cells are gonna die eventually if we don't do anything. So this area is called penumbra. And then this right in the middle is ischemic core. So this is a focal point of damage. Neurons cannot be rescued. And on contrast, this penumbra areas is the areas that surrounds ischemic core. And then these neurons are still alive, but they are in danger. So what can we do about this? So that's the whole idea of doing the ischemic stroke research, because that cell's done. You have to act fast. That's the only way you can rescue that. But what can we do to protect this penumbra so that we can minimize the secondary brain death? Just let me briefly talk about my research in a couple of slides. So I was interested in studying alpha-synuclein because when I started my PhD and then in ischemic strokes, I realized that, wait a second, the cell death mechanism of ischemic stroke is pretty much similar to Parkinson's disease. The only difference is that in Parkinson's disease, cells are dying in hours, in years and decades, whereas in strokes, cells are dying in minutes and hours and days. And I realized that, okay, if alpha-synuclein plays a critical role in controlling the brain death in Parkinson's disease, maybe it has some role in ischemic stroke. So I wanted to understand what's the role of alpha-synuclein in ischemic stroke. So first thing what I did is that I just collected some brain cells, plated in the petri dish, and then I did the ischemic stroke the experiments, put it in the chamber, just remove the medium, things like that. When I did that, so these bands basically represent alpha-synuclein. Darker band meaning more alpha-synuclein. OGD means just the cell culture experiments of ischemia. So when you see these alpha-synuclein expression levels, after stroke, you see more alpha-synuclein in the brain cells. So this was my first clue. Yep, people knew that alpha-synuclein can respond chronically, but it appears that within like a couple of hours, these cells are increasing alpha-synuclein inside of the cells. And this is more like a visualization. So when I stain these cells using the marker specific to alpha-synuclein, this is normoxia, meaning they're just control cells, no treatment, no damage. But once you subject these cells to these ischemic conditions, you see these green fluorescence dyes are all representing alpha-synuclein. So I was like, wow. So should I move on to the actual animal studies? So I actually did that. So I got some rats induced to ischemic strokes. What I did, I just kind of occlude using the silicon-coated filament, and then I physically blocked one of the brain vessels, which causes a massive brain infarct or ischemic cell death. So sham meaning nothing, just normal brain. MCAO is basically ischemic stroke. And then if you compare that, again, this band, intensity of this band directly correlates to the amount of the alpha-synuclein. I saw that within like 60 minutes of brain strokes, you see this massive increase of this alpha-synuclein. Okay, so from this kind of pilot studies, I kind of got the impression that, yep, so alpha-synuclein somehow acutely responding to this ischemic insult, even though the time frame is different. So my natural next step was, what happens if I just suppressing this increased alpha-synuclein in the brain? So I got this mRNA-mediated treatment. So what it does is that this alpha-synuclein siRNA is a small molecule that is specific to alpha-synuclein, this mRNA. And when it binds to that, it's just suppressing its protein expression. So you see less alpha-synuclein proteins. And then I was super excited for the success of this mRNA-mediated vaccines because my old study is done, so the therapeutic treatment is all done by this mRNA-mediated drug deliveries. And now it sounds like, I have an opportunity to move on to more like clinical trials. So when I did that, so this is control siRNA, so random sequence, not specific to alpha-synuclein, so it shouldn't do anything. And then these two conditions are ischemic conditions. So basically these animals are subjected to the ischemic stroke. So without any treatment, you see like increase of this alpha-synuclein. And then when you give this alpha-synuclein specific treatment so that you see less alpha-synuclein, first of all, I saw decrease of alpha-synuclein. By doing that, I know that my treatment works. So when I give this alpha-synuclein siRNA treatment, I saw less alpha-synuclein, and this is a quantification. So next, I saw molecularly I can suppress alpha-synuclein after stroke. What does that mean? If I have to see whether it is functionally improving these animals. So first thing I did is called Rotaro Test. So this is one of the tests to assess the motor function of these animals. So what I did is that I put these animals on this rotating beam. I think the beam is rotating at 8 RPM. So 8 turns every minute. So this is control animals, no stroke, just regular animals. And then this is a treatment. So these two animals are both received a stroke. So they are stroked animals. So these animals struggling a lot, right? They can stay probably like 10, 15 seconds, and they can't sustain, and then they have to just come down from their rod. But once you give this alpha-synuclein specific treatment, you can see this animal can go on and on and on. And then when I kind of plot the data in the graph, so 1 means 1 day after stroke, 3 means 3 days post-stroke, 5 and 7. So day 1 after stroke, the time, so this is basically 60 minutes, 120 seconds, 60 seconds, sorry about that, and 180 seconds and 240. So this is the time on the rotor, how long these animals stayed on the rotor. And day 1, not much difference, right? Day 3, there's some gaps between these two animals. Starting day 5 and 7, you see a dramatic improvement of these animals when I suppress this alpha-synuclein. The second test I did is I put this sticker on the animals' paws. And by nature, these rats, they don't like something on their paws. So if they're healthy and normal, they just remove it right away. As you can see here, this is control animals. And these animals, I believe, they are not having any strokes. So this is healthy. And as you can see here, as soon as we put this animal down on the ground, they just remove it. And then when you have ischemia, somehow you lose your function. First of all, your arms are weak, but at the same time, you lose the sensory functions. You don't feel this anymore. So even if they have something in their paw, they just don't recognize it. Of course, I couldn't talk to the animals, so I don't know what is exactly going on in their mind, but at least it appears that they just can't sense that. And then when you see that, this green line is basically the alpha-synuclein suppressing treatment groups, and then this blue line is the control groups. Got the stroke, but no treatment, basically. And then this is basically, y-axis is basically representing how fast these animals can remove the stickers, how long it took these animals to remove these stickers. And then if you get day 1 after stroke, so my cutoff was 120 seconds, 2 minutes. After 2 minutes, I didn't even count it. Starting day 3 and then day 5, as you can see, there is some spreading between these two groups. And then by day 7, those animals received alpha-synuclein suppression treatment. They were able to remove these stickers much, much, much faster. So I was like, I was really fascinated by this data. And then, more importantly, can we see a decrease in terms of brain damage? So pre means I gave the drugs before the onset of the stroke, because I want to make sure that this drug can target is inside of the brain so that it can protect the brain. Yep, when I quantify this brain damage area, it's smaller. But the real question is, can we protect the brain once they have a stroke? That's more clinically relevant, correct? So I induced a stroke, wait for 30 minutes, and then I gave this drug. And then I was still able to see this huge decrease in terms of brain damage. So again, after stroke, I found that alpha-synuclein levels are upregulated, increased inside of the brain. When I suppressed this alpha-synuclein, first of all, I was able to see a decrease in terms of brain damage, and they were functionally better in terms of motor recovery. I'm just going to quickly say that. So I also used the alpha-synuclein knockout mice. These are the mice that is genetically engineered so that inside of their brain, there's no alpha-synuclein. And I was curious, what happens to these animals if I induce the stroke? Are they going to do better, worse, or no change? That was my whole idea. If alpha-synuclein is really doing bad things, I want to see what happens if I just completely remove it. And then, first of all, this is survival graph. Green is alpha-synuclein, no alpha-synuclein in the brain. And then this blue is just regular mice, just normal mice with alpha-synuclein. And as you can see here, I gave rather severe stroke, a stroke that is too severe that it can actually kill the mice. So as you can see here, after day 1, yeah, both alpha-synuclein, with alpha-synuclein, without alpha-synuclein mice, they are pretty much like survival. Maybe 1 or 2 mice died in each group. But then as time goes by, day 3, day 5, and day 7, by day 7, if those mice with alpha-synuclein, 50% of them died. Whereas without alpha-synuclein, probably just total 1 animal's lost. So meaning that if I can somehow remove this alpha-synuclein, I can make these mice more resilient to rather severe ischemic stroke. And then I can also check the motor functions and measuring the brain damage. And then as expected, these animals actually show smaller brain damage and better recovery in terms of motors. And then when I checked these animals 3 months after, because what is alpha-synuclein? Originally, it came from Parkinson's disease, major component of Lewy bodies, forming the aggregation of these abnormal proteins. Can I see that? Strokes hugely upregulates this alpha-synuclein. Is it going to lead to formation of Lewy bodies? So that was my next question. And then, so I induced a stroke, wait for 3 months, and then I did this experiment. So proteinase K is the detergent. When this alpha-synuclein forms this abnormal aggregation and forms kind of this like protein clumps, they are detergent insoluble. When they are just regular, normal, just in a single forms, they are soluble to this detergent. So minus PK means without treatment to this detergent, plus PK means treatment was detergent. So this is opposite side of the stroked brain. Very little alpha-synuclein. Again here, after stroke, you see this massive upregulation of this increase of alpha-synuclein in both treatment group and then without treatment groups. Actually, this is treatment group without treatment groups. And one thing I noticed is that even with treatment of this detergent, you do see there's still lots of alpha-synuclein signals inside there. So meaning that these are detergent insoluble alpha-synuclein, which is the characteristics of those alpha-synucleins from the Lewy bodies. So the idea is that, yep, you have this ischemic stroke, and then based on if my research is true, you have an initial stroke, right? Somehow you have a stroke. And that increases alpha-synuclein inside your brain significantly. And then that somehow predisposes you to later onset Parkinson's disease. And in fact, there is some research going on that if you somehow induce a stroke in the striatal area, you can see early onset Parkinson's disease symptoms in these animals also. So I'm trying to say that, I know it's kind of still, the research is going on, but ischemic stroke, Parkinson's disease, probably it looks pretty distinctive to different like neurodegenerative conditions, but maybe they're not so much. And I think the key to the link to these two conditions is alpha-synuclein. Right, so that concludes my talk, and then this is my group right here, Dr. Vamganti right here, and then, and then my research is funded by the US Department of Veterans Affairs and American Heart Associations and NIH. Thank you for listening to my talk. Any questions? Yes?
Parkinson's is not fatal. However, it leads to a person not being able to swallow.
Oh, yes.
And they do swallow, they get pneumonia, they die of pneumonia.
Absolutely. So that's
kind of a false statement.
That is correct, yes. So it definitely has a complications depending on where which brain area is affected. And that complication can be mild, and as you said, like, if it affects your swallowing problems, they can cause the aspiration pneumonia, causing a serious infections, leading to death, yes. So even if I said that it doesn't lead to a fatal, it's not fatal, but in that sense, it can be definitely fatal. Yes?
You mentioned MSG.
Sorry about that. Yes.
Is that something that somebody should try to minimize?
I don't think so. So the reason why as a researcher, I never say a statement very definitively, but I can say it's pretty safe because MSG is one of the most hated kind of condiments on earth. And there's so many people, want to hate this and they want to shut down the whole, manufacturing of the MSGs. And there's so many targeted research, trying to prove that this MSG can cause brain damage and the excitotoxicity that I talked about. But there's no conclusive data so far at this point. There's no one data showing that, yep, if you give this MSG, you can damage your brain cells. And there's no such data so far. And there's so many, FDA studied that for it, and then there's so many private labs studying for MSG. There's no data for it. So my conclusion is that it's probably fairly safe. And then it's been out more than one generation. And so probably at this point I can say it's safe. But of course, we don't know what is exactly going on, so it's hard to say.
Well, thank you.
Yeah, any questions? Yes.
Is there currently a treatment used for humans to prevent that, the toxic chemicals right after a stroke when they start to spread to the other cells? Is there a treatment to prevent that?
So right now, the only treatment we have is this called TPA. So which targets those blood clots. So you probably heard about the term clot busters, right? So we do have that. We do have this mechanical thrombectomy, meaning that we just insert this device, just physically grabbing that the blood clots and take out if the clot is formed in like big vessels. If it is formed in the smaller vessels, we can't do much about it because it's just too small. So mechanical thrombectomy and these clot busters, there's no neuroprotective agent at this point. But there's lots of research is going. In fact, I think the NIH is spending annually about like $70 billion for medical research. So hopefully, yeah, we can find something in the future. Any other questions?
So if someone who's having strokes,
Yes.
small strokes, and become more and more common, what's happening? What's happening? Why is he getting more and more strokes? Is this getting worse and worse?
Are you saying that it initially starts with like TIA, like mini strokes?
Minor stroke. I'm not sure what the word is.
Mm-hmm.
It's a minor stroke, and then it just had more and more strokes.
Okay.
That were similar to that, but each one was worse and worse.
Right. So that's because of some of the risk factors I talked about, like cholesterol and like hypertension, diabetes. So it's hard to say what exactly causes like progressively worsening episodes of strokes, but what we can say is that like because of that, like doctors are trying to minimize the future occurrence of the strokes. So that's why like we're like paying so much attention to controlling your cholesterol levels, and because strokes, if it is ischemic stroke, it's gotta be coming from somewhere. It's either coming from the cholesterol, those blood clots, or it's coming from somewhere else. Our bodies form somewhere else, and then it lodged in your brains. So if we can somehow decrease these cholesterol levels, because we know that these blood clots, the major component of this, the cholesterol. So controlling cholesterol can be a really significant. That's why doctors' favorite drug is a statin, by the way. So it's quite safe. Minor side effect, which is like muscle pain. If you can tolerate it, one time I did some 10-year risk of ischemic, the overall cardiovascular risk for one particular patient. With or without statin, you can reduce the risk of having a stroke by half, which is like really significant. And then it's not like from 2 to 1%. This patient without statin is like 24%. By taking statin, you can reduce that to almost 10%. So that's why we really pay attention to stroke, the cholesterol levels. And then if it is more like atherosclerotic ischemic stroke, that can be caused by a rupturing of one of those atherosclerosis, and that can be caused by high blood pressure. So that's why doctors are saying that please take your blood pressure medications. Are you taking it? What's your home blood pressure? Controlling your blood pressure is also very important. And then diabetes, that can double, triple your risk of having a blood pressure. So controlling your A1C level is also very, very important. So all those, all those preventive measures can potentially lower the future risk of having a stroke.
Just to add on to that a little bit, when I think of strokes, there's no one coherent cause. It really depends on who you are and what comorbidities you have. So Tehi kind of touched on some of the things that can cause stroke. High blood pressure can lead to plaque buildup in your vessels. Some of those plaques can shoot off and occlude those vessels like we talked about. You can have diabetes, which can do the same thing. You can have kind of things building up in your carotid arteries that can pinch off the blood supply to the brain. If you have a vegetation in your heart, that can shoot off little chunks of whatever it is to your brain. So when you talk about why strokes may progress from mini strokes to bigger and bigger things, it's a it could be a destabilization of whatever disease you have. So for example, if you have a vegetation in your heart and you don't get that under control, at the beginning when the vegetation is smaller, it's throwing off smaller clots. So they get deeper into the brain, deeper vessels, they only pinch off little vessels, cut off that small amount of blood supply. But if those vegetations get bigger, for example, then they're blocking the bigger vessels. You're trying to fit a bigger, whatever it is, into a tube. You're going to get pinched off quicker if the vegetations are bigger. So that sort of thing, just like destabilization of whatever is causing the strokes in the first place, can lead to bigger and bigger or more frequent strokes.
I've heard that serious concussions, like an accident or whatever, can cause some of these similar problems years later. What's happening in that situation?
You mean concussion causing,
Just banging of the head.
like what symptoms are you specifically mentioning about? Like stroke types of symptoms or? So concussion can cause later onset like headaches and like fatigue, things like that. And again, this is trauma to the brain, right? So it can cause microbleeds inside of the brain and rupturing of some of the blood, and then maybe also predispose some of this pro-inflammatory molecules. And then, in fact, there is a research going on for this traumatic brain injuries. And then when these animals are subjected to this traumatic brain injuries, they tend to have more this pro-inflammatory molecules, which predisposes to all sorts of like brain diseases, like later on diseases like Parkinson's disease. I've seen that traumatic brain injuries and wait for 3 months, and then these animals can show some of the movement disorders that is similar to Parkinson's disease and cognitive deficits based on some of this cognitive functioning testings like Morse water maze and things like that. So these animals can have long-term consequences based on the severity of the traumatic brain injury.
Is the question, are you referring to like CTE in football players and why they get problems?
Yeah, the football players and things of that sort.
Yeah, it's, I would say it's more of like a progressive accumulation of problems over time. So in football, you're hitting your head multiple times as you go through plays, get tackles, that sort of thing. So you're just kind of repeatedly bludgeoning your brain, basically. It's kind of rattling around in your head. That accumulates over time. So I would say if you have one concussion, you fall off your bike, hit your head, provided it's not a major problem, I would say you'd be pretty much okay 10 years, 20 years, 30 years down the road. But if you have this repeated trauma like football players have, that's what's causing the problem.
I feel like it all depends on the severity of the conditions also. Like Aman last week talked about this brain reserve. So once you deplete that brain reserve, then that's when you actually have all these sorts of problems. So if it is mild traumatic brain injuries, maybe the later onset symptoms are pretty mild and maybe even less. But it can be different based on the severity. Any more questions? All right, thanks for coming. And I know I was planning to do about an hour talk, but then it was way over like an hour and 30 minutes. Thanks for bearing with me. Thank you.